Neuroscience on deck. So he is an associate professor of chemical and biomolecular engineering at NC State University. His group engineers cellular and molecular platforms to understand how information is stored and accessed in biological systems. Please welcome our first speaker, Dr. Albert Keung.
All right, thanks everybody. It's great to be here. So I'm going to start with a decidedly non-science story that often pops into my head actually when I'm thinking about a new project. So about 10 years ago, I was doing a postdoc up in Boston, actually where my parents still live. I was having dinner with my parents, and my mom had just made a really delicious dish. It's kind of eggs hard-boiled in a soy sauce-based broth. And she was recounting, she was saying, oh, this is your dad's favorite dish. And I looked at my dad, and he looked at us a little sheepishly and said, maybe after 30 years of marriage I can say this, but I don't really like eggs. And my mom was like, what? But you always eat it so quickly. For like from the first time I made it for you until now, you always eat it really, really fast. He's like, well, the first time you made it for me, we were dating, and I wanted you to keep dating me. And then after that, somebody you love makes you something to eat, you eat it. So anyways, it's not science related, except that it pops into my head a lot because observation is really useful and important in science, right? But we can run into some quandaries with correlation. Basically, in this case, my dad always ate these eggs really, really fast, but you can come up with several different conclusions from that, or interpretations. One is that he really likes the eggs, or another interpretation is he really loves my mom, right? So this gets me thinking a lot about approaches in our group because the human brain is super complex, and it's very hard to study. We can observe it in postmortem tissue. After somebody dies, you can take the brain and slice it up, image it, study the anatomy. There are some really advanced non-invasive methods to image and study the brain now. And those have given us tremendous insight and continue to be super important. But it'd be really nice if we also had a complementary method where you could experiment, right? Perturb, modify, and see how parts of the brain respond in a molecular and cellular level. So that's what I'm going to tell you about today, kind of a new model that I think will be complementary to a lot of the really advanced imaging techniques that we have today. So you can take cells, skin cells or blood cells from an adult person, and you can reprogram them into something called stem cells. And you can use what we call reprogramming factors to do this. And these stem cells can then make those little tissues over there called human cerebral organoids. And I put a little penny there to give you some sense of scale, so they're usually a few millimeters in diameter. Now, what are great about these are that they're pretty easy to make in the lab. You can generate them from adult people with different types of diseases, different backgrounds and genotypes. And they contain, if you slice them open and zoom in and stain different cells with different colors, you can see that they have a lot of different cell types, so over tens of different types of neurons, astrocytes, progenitor cells, and they can form some polarized type structures as well that are reminiscent of the type of structures that you see in their brain. The other advantages are that they don't think, right? They're not making neural connections at the level that you can, that it would be concerning ethically. And so you can really use these models as experimental systems to perturb and study how they react. So I'm just going to give you a few different examples of different types of perturbations that you can do with these and what you can learn from them. So actually this is a project that an undergraduate and a PhD student in the group collaborated on, and the undergrad really liked kind of mechanical things, and so she asked, well, what happens, are your brain cells and tissues sensitive to mechanical perturbations? Oftentimes when we think about biology, we think about soluble factors and things floating in solution, but we're physical creatures, right? And we experience forces. And so she created these microwells out of these hydrogels, and then she and Delara forced these organoids to grow inside of them in different shapes. And what they actually found was that if you grow them in shapes that have more constraints, more kind of points of indentation and strain, these organoids will have cells in them that are biased towards turning into one type of neuron over a different type of neuron. Another graduate student was interested in substance abuse disorders, addiction. And so he asked, well, could these organoid models tell us, or at least recapitulate some properties of addiction that we know exist in rodent models, for example. And so what he did here was he took some organoids, he exposed them to dopamine, which is the neurotransmitter that is regulated by cocaine predominantly. And he would expose one batch of organoids to an acute dose, so one single dose of dopamine. And he saw several hundred different genes turn up and down on basically change in their levels in these organoids. And then he also exposed a different set of organoids to dopamine, but in a repeated fashion, chronically over a week. And what he found was that if you do it chronically, the number of genes that respond to dopamine decrease. This is called gene desensitization, and it actually kind of mirrors in a kind of macro scale level what you might expect to see in a model of addiction, right? If you continue to take a drug of abuse, then you might see your response to it decrease over time. And then the last example I give you is you can create organoids out of cells from people with different types of diseases or disorders. So our lab is particularly interested in a neurodevelopmental disorder called Angelman Syndrome. And it occurs in about 1 in 15,000 children. And in that disorder, UBE3A is a protein that's lost in neurons in the brain. And so you can see that on the top left where the red staining is very dim. Over at UNC Chapel Hill several years ago, they actually discovered some chemical compounds that could turn up that level of the protein. It's called indotecan here on the bottom. And so we asked, could these organoids be used to screen or test potential therapeutics, things that could turn back up this gene that's lost in kids with Angelman? And indeed, you can see that when you add this chemical compound, you can see this rescue in the level of this protein. The neurons in these organoids are also electrophysiologically active, so they're firing action potentials. And in patients with Angelman, these are hyperactive, and you can see a lot of, it's kind of like an EKG signal, if you guys are familiar with that. So you can see a lot of hyperactive spiking in these types of neurons, in these organoids, and if you add this chemical compound, you can basically calm that down. So to kind of bring this back 180, the human brain is very challenging to study. I think what we really need is we need all of those powerful imaging and non-invasive techniques, and we need to continue advancing those. And it would be really nice if we have these complementary models that we can also probe at the molecular and cellular level. And with that, thank you for your time. Happy to take any questions.
Question. I see a hand right here.
You talked about the chronic effect of the more it's exposed to something, the less effect it has. Can you talk about how that plays into changing habits? If you think about from a pain perspective, right, there's TENS machines that send electrical signals to the brain to kind of overwhelm the brain so it doesn't think about the pain anymore. How does that relate to changing habits?
Yeah, so in this case, we were specifically looking at organoids that kind of model the reward center, so the striatal areas of the brain. And what happens in the case of cocaine, for example, is that you basically get too much dopamine. And the cells have certain certain kind of gene responses that they'll have in response to normal levels of dopamine. But if you have too much dopamine due to cocaine, those responses get tamped down over time. And so you need more dopamine or more cocaine in order to get that same level of response. So that's part of the addictive aspect of it. But fundamentally, you're actually changing kind of the threshold at which your neurons are responding. And that's, I think, a major problem underlying kind of molecular cellular mechanism underlying substance abuse.
See a question right here.
So I heard you say that there were the samples you're taking for stem cells do not form the neural connections. So I'm wondering if you have been attempting to sample brain cells and glial cells. I don't know if your research is moving in that direction, or how are you mimicking the brain part of activity in your research?
Yeah. So this is there's ethical considerations that we have to have. So we think about that a lot, and how far do we want to advance these models, right? So there's work where, including in our group, where we're interested in fusing organoids that kind of mimic different parts of the brain to try to understand those connections and how they interact. And so you often see maybe like 1 to 3 levels of neurons kind of connecting. But these things have, 1 million, 10 million cells. The human brain has 100 billion, so over 3 orders of magnitude difference. So we can't ever recapitulate, the complexity of the human brain. But we can start to, I think what we can use these things for is to study kind of levels of 1, 2, 3 connections, neural connections, but not, the level that really you need to study like true behavior. Yeah. So there's definitely limits to what you can do with this.
I see 2 questions. We'll get to you both. First one right here.
So in the case of addiction, different brain regions are differentially impacted by drugs of abuse. So do you have the ability to differentiate these stem cells into neurons that would be present in different brain regions?
Yeah. So there are different types of organoids. So you can make forebrain organoids, midbrain, hindbrain, dorsal, ventral, organoids. So you can do that. The other thing you can do with a lot of the upcoming recent omics technologies that you can actually study. You can take these organoids, and they contain about 30 different types of neurons actually in them. Many different subtypes of neurons. And there are methods where you can study the gene expression of individual cells. And you can do that for hundreds of thousands of cells. And so we actually have, we've done that with cocaine, with morphine, with a few other compounds to study how individual different subtypes of neurons respond differently to drugs of abuse.
Last question right here in the front.
Hello. So I'm curious if you've studied brain development, considering the work that you were talking about regarding the organoids and how physical stress can change differentiation, alongside, say, using some chemicals. Has your lab done work on brain development and using organoid differentiation in comparison to brain development, or are the 2 subjects just too different to study by comparison?
Actually, that's a great question because development is actually, neurodevelopment is probably the best aspect of human biology that these models model, right, can capture, because they tend to capture earlier stages of development rather than adult, right? So a lot of the addiction work that we're doing, it's hard to translate that directly because they're not as mature neurons as they are in the adult brain. So the neurodevelopmental aspect is really key, and people have done things where they've compared like the characteristics of these cells with early neurodevelopment in humans, and they found a lot of similarities, basically. And one of the useful things that we found is with Angelman Syndrome, one of the key questions in that disorder is, when do you need to treat the kids? So if we develop a therapy, when is treating them at 1 year old enough? Sometimes we don't diagnose children early enough to be able to treat that early. So one of the things that these organoids showed us was when this protein that is lost is actually lost, and at what point can we reactivate it. So we actually use these models to figure out kind of that therapeutic time window at which therapeutic treatment might have the most impact. So definitely neurodevelopment is important. Yeah.
Thank you so much, Dr. Albert Keung.
Thank you.
Great job.